Now we will be continuing in the lecture related with the synthesis of collagen. We have already discussed that collagen molecule is basically a protein, and it's the most abundant protein in our body, and this protein is mainly present extracellularly. It's one of the major component of connective tissue. Is that right? Collagen fibers. Now, as I told you previously that collagen molecule is basically a triple helix of 3 peptides, right? We have multiple genes which can make different types of alpha peptides, and those alpha peptides can be made into triple helix so that collagen molecules are made. Now we go step by step that a fibroblast which got right now instructions to synthesize and secrete collagen, right? How it starts. The step number 1 should be that it should open its DNA. You know, DNA is tightly double helixed. It should open a part of the DNA which is actually having the gene for the alpha peptide. Is that right? Alpha peptide means that alpha peptide which is eventually going to be a part of collagen molecule. Now, for example, this is alpha 1 collagen gene. Now, this has been activated. Once this gene is activated, then step number 1 must be that from the DNA we should transcribe messenger RNA. So, what is the first step here? Transcription, right? That is transcription. What is transcription? Yes, transcription is a process in which DNA coding is transcribed into RNA coding molecule, right? Then once the messenger RNA is made, right, originally it is made pro-messenger RNA, and this pro-messenger RNA undergoes some, suppose this is the messenger RNA, it undergoes some modifications within the nucleus. After those modifications, this messenger RNA molecule will come out. Now here is the messenger RNA molecule. Now, this messenger RNA molecule has the nucleotide sequence which will direct the synthesis of peptide chain, and that peptide chain eventually will become the part of collagen molecule. Is that right? Now, this messenger RNA will be arranged on ribosomes. What is this? These are ribosomes. These ribosomes are attached on, yes, what is this? Endoplasmic. You know endoplasmic reticulum? What is endoplasmic reticulum? It's a set of membrane-made tubes within the cytoplasm. This is present in every cell. Every cell has a membranous tubes within its cytoplasm, and this network of tubes within the cytoplasm, it's like a network, you know. For example, this is endoplasmic reticulum. It's a network of tubes. Now, this network of tubular structure within the cell is called endoplasmic reticulum, right? Actually, this plays a very important role in protein synthesis, right? Why? Because ribosomes are going to synthesize proteins, they get attached with the, what is this? On the surface of endoplasmic reticulum. And that part of the endoplasmic reticulum which is loaded with the ribosome, because under the electron microscope it was looking rough, so it was called rough endoplasmic reticulum. And this is an endoplasmic reticulum which is not having ribosomes, so it is called smooth endoplasmic reticulum. Now, collagen is a protein. Is that right? It has to be synthesized by the ribosomes. So, of course, its synthesis will start on the rough endoplasmic reticulum or on the smooth endoplasmic reticulum? On the rough endoplasmic reticulum. So, what really happens? that first of all DNA is transcribed into RNA, and then RNA, messenger RNA comes out, binds with the ribosomes on rough endoplasmic reticulum. After that, process of translation start. Translation process means that messenger RNA sequence of nucleotide is translated into sequence of amino acids in the peptide chain, right? Of course, you already know that transfer RNAs will come here and they will bring amino acids, and then ribosomes will link the amino acids with each other, right? So what really happens that, let's suppose we start here, that as messenger RNA, for example, this is ribosome set number 1, this is ribosome set number 2, this is ribosome set number 3. Now, actually, it is one translator, it is second translator, it is third translator. Every translator will read all the messenger RNA. It means every translator will make its own peptide chain. For example, this was the first one, this ribosome set was the first one, and messenger RNA started sliding from there. By the time messenger RNA, of course it's a long one, this ribosome, messenger RNA is moving in which direction? Into that direction. So this ribosome has read the translated the maximum one. Again listen, this is to be translated, right? This is translator number 1, this is translator number 2, this is translator number 3, and at one time with one messenger RNA the multiple ribosomes. Is that right? Now, actually I made the direction wrong. This direction should be in this direction if it is moving like this. If messenger RNA is, this is like this, so it is moving in this direction. Is that right? Now when it is moving in that direction, this has read the maximum part of it. Suppose this is the early part of gene, this is the middle part of the gene, middle part of the messenger RNA, this is the later part of messenger RNA. Is that right? So this has read this part, and this part. So it has read the maximum part. It has read the little less. It has translated a little less. So every translator will be translating this code into amino acid sequence, but this has made the maximum amino acid sequence. It has translated a little bit less. It has translated much less. But as it will keep on slipping, every ribosome will reach all the information, and eventually when, let's suppose, there is a ribosome here, as it is moving in this direction, there is a ribosome here. It has read all of this, it is at the tail end. It means this ribosome set has read most of it. So it means it has translated into longest peptide chain. Is that right? And by the time ribosome reaches to the end, they fall away. They fall away. These are assembled ribosomal machinery disintegrate. It fall away, disassemble, and peptide chain is released. Am I clear? No problem into this? Right? So as messenger RNA is moving through the ribosomes, they keep on translating. So from this one, peptide chain will come, then this will make another peptide chain, then this will make another peptide chain, and this will make another peptide chain. So it means if one messenger RNA has 10 ribosomal units, so it means at a simultaneously it is being translated into 10 peptide chains, but every peptide chain will have the same amino acid sequence. Do you get it? Yeah. Am I clear? Right? The only difference is the ribosomes which are most near to the tail end of the messenger RNA, they must have translated the most of it, and which are near the head end of the they must have started translation just now. Is that right? But eventually every ribosome will translate the same amount of messenger RNA and produce the same length of peptide chain with the same sequence of amino acids inserted into that chain. Am I clear? So what really happens?
That's translation, right?
That is translation. That's translation. So if it is moving in this direction, so peptide chain, now I will do here it is maximum translated, here it is less translated, here it is least translated. Do you understanding?
Yeah.
Yeah.
Quick question. These peptide chains are these the same peptide chains that are the alpha chains? That
Yeah, these are alpha chains.
Okay.
For example, if this is alpha 1 chain, this is also alpha 1 chain, this is also alpha 1 chain. Because it means that if with one messenger RNA, there are 20 ribosomes, it means 20 alpha 1 chains are being synthesized simultaneously.
Right. So they're all the same alpha chains, but what about the other two?
Other two, yeah, now look. For example, if the collagen we are going to make, let us suppose the collagen which we are going to make will have one peptide alpha 1, let us suppose just, and should have another peptide which is alpha 2, and should have one more peptide which is alpha 3. It means to synthesize such a molecule, 3 genes should be activated. 3 genes should be activated, 3 different types of messenger RNA should be made, right? And 3 different types of peptide chains will be made. All of these peptide chains are alpha chains, but they are slightly different from each other according to amino acid sequence. It means to synthesize collagen molecules with different combinations, there are multiple genes present in our genomic setup. Am I clear?
Yeah.
Sure? Okay, now come back. So, for example, we are just making alpha 1, right? Procedure for all other is the same. So, we just concentrate how alpha 1 is made, right? Or now, this alpha 1 is being synthesized. Now, this chain which is initially synthesized, right? What are these? Amino acids, right? And this chain is called pro alpha peptide. So when they are freshly made, what are they called? Pro alpha peptide. Any question here? Now, every chain when it is made they have a very special type of amino acid sequence here. These are called signal sequence. These are called signal sequence. These signal sequence will not be the part of final molecule, but they have a very important function. Have you seen sometimes there's a needle and there's a thread, and you want to, for example, you want to do some thread work, you need a needle. But do you think in the end, once you have completed your thread work, needle is removed or needle is there? Oh, you have good general knowledge. Needle is removed. Same is true. This signal portion of the peptide chain is the needle, which will guide this peptide thread. Guide this whole peptide thread to enter into endoplasmic reticulum. Because it is being synthesized, this peptide thread is being synthesized on the surface of rough endoplasmic reticulum by the ribosomes, which are translating messenger RNA into peptide chain. But this signal sequence, this is also amino acid. This small sequence acts as a needle. If this needle is there, only then this peptide chain will be allowed to enter into endoplasmic reticulum. How come? Let me explain. Let's suppose this was the peptide chain which was being synthesized, and now this is the needle sequence. Is that right? Now this is called signal sequence. Signal sequence of amino acids. Actually, in the endoplasmic reticulum, there are very special type of proteins which can recognize this. So as soon as this signal sequence gets stuck with it, this whole thread is pulled into the endoplasmic reticulum, as needle is going and pulling the thread with it. You are understanding?
Yes.
Sure? Right? So, again, up to now, how many steps we have learned? Number 1, DNA, gene for the alpha peptide of collagen, transcribed into messenger RNA, then messenger RNA come into cytoplasm. This messenger RNA binds with the ribosomes on the rough endoplasmic reticulum. There this messenger RNA is translated into pre-pro alpha peptide. Pre-pro alpha peptide has signal sequences. These sequences help this peptide chain to enter into endoplasmic reticulum, right? So what really happens that here is, let's suppose that channel group of proteins, okay? We'll just write down here. These are the group of proteins, and as soon as signal sequence gets stuck into this, right, and these are the other amino acids, then this will come into this. In this way, this peptide chain, this is signal sequence, and what are these? Amino acids, right? Now what happened? That this peptide chain, which has been synthesized, now has entered into endoplasmic reticulum. Here, there is signal chain is disintegrated, cleaved away. Thread has come into its position. We don't need the needle. So there are enzymes in which digest away the signal sequence. Is that right? After that, what is left behind is called, now what is left behind is called alpha. Initially it was pre-pro-alpha peptide, now it is called just pro-alpha peptide. Is that right? So it was synthesized as pre-pro-alpha peptide, and pre portion is actually signal sequence, and it drag all the chain into endoplasmic reticulum, and in this special tunnels, we'll do special therapy to this molecule. Right, we'll talk about that. Why we have brought this molecule into this tunnel system, into and the endoplasmic reticulum, there's a purpose in that. But once it comes in, signal sequence is digested away, and remaining molecule is called pro-alpha peptide. Is that clear? Now, let me tell you the sequence of amino acid, the basic principles of sequence of amino acid in collagen peptides, alpha peptide. The basic sequence should be, and most of this is one amino acid, right? And okay, I will make it more. One amino acid, another amino acid, another amino acid. Then one more amino acid, another amino acid, another amino acid. Can you see this thing? That this amino acid is appearing again and again. This is glycine. What is it? Glycine. Actually, the whole alpha chains are having special sequence of amino acid that every third position is glycine. Again, glycine, X and Y position. Is that right? So basic formula of this is glycine, X, Y. And this sequence keep on repeating throughout the thread. It means the nucleotide sequence in DNA, which is coding for glycine, is appearing again and again. Nucleotide sequence, which is coding for glycine, is appearing again and again in DNA. So again appears again and again in RNA. And when the peptide is translated, glycine appears again and again. Right? So every third position, what we have? Glycine, glycine, glycine. You may be thinking, why glycine is there? Do you really want to know, or we just continue our lecture? You want to know it. It's very important. Actually, in future, we'll take 3 chains and twist them. In the future, we'll take 3 chains. This is suppose one chain. This is another chain. This is another chain. And we have to make a triple helix. Actually, on every third position, there should be glycine, glycine, glycine. Glycine is a type of amino acid which has a very small side chain. It is a very, very small side chain. Is that right? Now, when you will twist them tightly, because they have a very, very small side chains, they fit with each other very tightly. You just imagine 3 very smart people hugging each other, or 3 very fat people hugging each other. Where will be really true hug? You have experience, yes. Small. What? Smart people or small people? Smart. I mean, if some very fat people with very big amino acids with very big R chain side chains, when they try to come with each other, they just bump around. They don't really, right? So, nature is so wonderfully working. It's not just everything by random. That collagen has to be very tightly twisted. It provides the real strength to our body. If there's no collagen, if I don't have collagen in the skin, I'll just do like this and skin will peel off. Is that right? So because it has to provide the real strength, so nature wanted that when three alpha chains twist with each other, they twist tightly. For every third position, nature has put glycine, which is the amino acid with smallest side chain, a very smart amino acid. Is that right?
It's the smallest of the 20, no?
Yeah.
Adenine, I thought, was the smallest side chain.
This side chain is so close with each other that it's twisted tightly. Now, it has a clinical point with it. What is the clinical point? That there is a disease in which, listen now carefully, there is a disease in which the nucleotide position which code for glycine, that is mutated, that is altered. There is some inherited disease in which the genes are defective. What is the defect in the gene of the collagen? That the point where there should be glycine, that is altered, and the places where glycine should be there, at every third position there should be glycine, isn't it? Every third position there should be glycine. So, if due to some reason the point which is coding for glycine is altered, then altered or mutant code may not now code for glycine. Maybe some other bulky amino acids are fit into these positions. And do you think then collagen can really tightly twist? So they will make tight triple helix or loose. And when they will make loose, collagen will gain its strength. And when collagen will not gain its strength, do you think it will be weak or strong? Weak. And in these people, they make their bones. Bones have a lot of collagen. Is that right? Their bones are so weak that even within the uterus of the mother, with the pressure of uterus, the bones of the fetus are crushed. And in very severe cases, there is intrauterine death.
Quick question. You said the reason for that is because of their genes not having a strong...
Look. If you alter this, for example, here is triple C. That is altered into CGC. So it means that point become mutant. So triple C was, let us suppose, coding for glycine. And CGC, which is altered, is coding for some other amino acid which is very heavy side chain. Can it snug properly? No. So collagen will achieve its strength or not?
So there'll be like one point out of the chain that's not...
Yeah.
Okay.
And collagen will be very weak. And if collagen is so drastically weak, right, if so drastically weak, then bones will be very, very weak. And if bones are very weak, some of the some of the fetus, they undergo multiple fractures before even birth of the baby, right? And in some milder cases, babies are born, but baby just fall from the sofa and get ribs fractured and legs fractured and arm fractured. Is that right? That is not Marfan syndrome. That is not Marfan syndrome. You have seen Marfan syndrome, they are tall people, and that is adult onset problem. In this case, do you think bones are made perfectly or imperfectly? So osteogenesis means formation of bones, is imperfect. So disease is called osteogenesis imperfecta. What is the disease called? Osteogenesis imperfecta. Osteogenesis imperfecta. That osteo means bone, genesis means formation. Bone formation is imperfect. Is that right? Because of mutations in the genes coding for the alpha chains, especially for that collagen which is present in the bone. I told you the many types of collagen, right? These type of mutations which lead to this disease, these mutations should be in those alpha chains which are responsible to make collagen for bone. Which type of collagen is present in bone? Yeah, there is so many types of.
Collagen type 1 and 3.
There are so many types of collagens. Which collagen type is present in bone? Do you know spelling of bone? Do you know spelling of bone? Please tell me. B-O-N. Collagen 1. So when there are such type of mutations in those alpha chains which make the collagen 1, in which chains glycine insertion in nucleotides in the peptide sequence in alpha chain is disturbed, or at many points glycine is replaced by more bulky amino acids, then triple helix cannot be tightened well and the end result will be very weak collagen. Of course, degree of mutation in different people may be different. Some may have very severe problem if more glycine points are disturbed. Some may have moderate problem. Some may have mild problem. You could not understand. Look, there are multiple genes which make these chains and there are multiple points for glycine. So someone has problem at suppose 5 points. Other person has problem only at 2 points. Another person has problem in 1 point. So who will suffer most? 5 points mutation. And person with, so people may have severe problems, severe type of mutations, or mutations leading to moderate problems or mild problem. It means osteogenesis imperfecta is not one disease, it's a whole group of diseases, it's a whole group of problem, right? For example, someone, product of conception in which these genes have multiple defects and they make very, very weak collagen 1. Then fractures will occur within the uterus before the birth, and even skull will be fractured, ribs will be fractured. Do you think brain of the fetus or lungs of the fetus and heart of the fetus can work well? So there will be intrauterine death. That so severe problem is called osteogenesis imperfecta congenita. Yeah, that is fascinating to learn and sad to have. Congenita. Opposite to that, congenita means congenital. Congenital means someone born with a defect. But someone has a moderate problem, right? That baby will be born, the defect is compatible with life, but that baby who is born with osteogenesis imperfecta with moderate problem, with little bit trauma, he gets multiple fractures. That is called osteogenesis tarda. Tarda, right? These type of babies, parents are under big trouble. You know why? Number one, their baby is suffering, of course. That's very easy to understand. That for example, mother is holding the baby, right? He just hug him tightly and 2 ribs are fractured. It's very sad, right? Baby just fell on the ground and got his femur fractured. This is one problem. Second problem, when parents take these babies to the hospitals, usually hospital people have because this condition is rare and child abuse is more common, so hospital doctors, they suspect that probably baby has been battered and beaten. So they call the social service people, and all the time the parents are in trouble to explain that we are not the culprit. Eventually some good doctor will make the diagnosis of osteogenesis imperfecta. And when parents will ask, what is this disease? The doctor will say that in simple word, we call this disease brittle bone syndrome. Brittle bone syndrome. So what we have done right now, that at molecular level, the defected DNA was the mutation or defect in DNA was transcribed into defect into RNA, eventually lead to faulty translation and faulty alpha chains, which could not make a proper triple helix and lead to reduced tensile strength in the collagen fibers, translating into brittle bone disease. Am I clear? No problem. Let's come back. We are still in the beginning that how to make it. But I told you that glycine to have at proper position is very, very important. Then what happens? That as this pro-alpha peptide is moving through the endoplasmic reticulum, there is some very interesting points, and here there is an enzyme. Here there are two enzymes here. Whenever they find pro-alpha peptide moving through this, right, they do a modification there. Of course, you know that this is glycine, and I did not tell you what are the amino acid as position X and Y. Usually at glycine position is by glycine. Suppose this is glycine, right? In these positions, especially Y position, you have proline or lysine. What are those arrangement there? Proline or lysine. Now, it means that throughout the chain, glycines are appearing very regularly. Moreover, in these collagen forming chains, there are a lot of prolines and lysines. Is that right? Now what happened? At this position, what are these? These enzymes put special hooks, biological hooks. What are the advantages of these hooks? So these chains will be eventually, when they are formed, not only they will twist, they will hook with each other tightly. You get it? They will hook like this. Now what are these hooks? These are actually hydroxyl points. There is hydroxylation of prolines and hydroxylation of lysines. If this hydroxylation is not done, we are again in trouble. I will tell you later. These two friendly enzymes are responsible to do hydroxylations of at multiple points. What are these hydroxylations of prolines and lysines? So in this way, in pro-alpha peptide, lot of specific prolines and lysines are hydroxylated by these enzymes. So what is the name of these enzymes? Hydroxylases. The name of these enzymes should be hydroxylases. One should be propyl hydroxylase and other should be lysyl hydroxylase. Is that right? But both of them are in love with a very special lady. What is that lady here? Until they keep on having that lady, they keep on working. Yes, who is that lady? Yeah. If these enzymes are having this lady in between or with them, they keep on working and keep on putting the hooks on alpha peptide and keep on making it hydroxylated. What is the name of this lady there? You have heard that, I think there's some sentence like this, that behind every successful man, there is a woman who has her hand in his pocket. And other women are thinking how to get a chance. I think you have not heard the second part. Okay. Men are decent, they don't mention the second part. Okay. Anyway, so I think these enzymes have also something like that behind these enzymes there's a lady, right? And until that lady is there, they are hydroxylazing the alpha peptides, and these enzymes are doing hydroxylation at proline and lysine residues. So these are called propyl hydroxylases and lysyl hydroxylases. About this lady, you must know, you are doctors, or going to be doctors. What is this? This lady is like a cofactor for these enzymes. This lady molecule is vitamin C. What is this molecule? Vitamin C. You can get these molecules from citrus fruit. Is that right? Like, what are citrus fruit? Oranges. Not banana? Okay, right. Orange, grapefruit and things like that. Right? So many fruits which are called citrus fruits, they are very rich in vitamin C, ascorbic acid. Vitamin C especially goes into, it does many functions. One of its very important function is it goes into fibroblast. In that they goes into endoplasmic reticulum and they develop very special relationship to stimulate these hydroxylases. So as peptide chains are being made, they get hydroxylated. Now, let's suppose this lady is not there, vitamin C is not there, these enzymes will become very sad and depressed, they don't work. And if they don't work, then what happens? Chains are still produced without hydroxylations.
So they only work when vitamin C is there.
Yeah, it's like you. Right? These enzymes only work.
I don't know what he means by that.
Yeah. These enzymes only work when vitamin C is there. They are called ascorbic acid dependent enzymes. Right? Ascorbic acid is their cofactor. They are co-performer. Now, when these enzymes are, when vitamin C is not there, these enzymes are not working. Do you think collagen will be produced? It will be produced, but it will not have hooks. Because production is dependent on different machinery. Try to understand it. It will be produced, genes will still open. When you don't eat vitamin C for a long time, genes will still work, messenger RNA will be still there, ribosomes will be still translating, peptide pre-pro-alpha peptide will come, convert into pro-alpha peptide, but it will not get hydroxylated at proper propyl, proline and lysine residues. Is that right? When they don't get, what is this? Hydroxylated, when this alpha chain is made, this alpha chain was supposed to have these hooks. These hooks are not there. They will still twist, but loosely or tightly? Loosely. And even not only triple helix will be loose or weak, even when multiple triple helix come together to make a larger fibril and fiber, even that will be loose arrangement. So collagen will be strong or weak? Weak. Right? And in these people, collagen, why your teeth don't fall out? Because you are a young man. But more important thing is because you have very strong collagen there which anchors the teeth with the gums and bones. Is that right? Yeah. So what happens? When you have deficiency of vitamin C for a long time, then what really happens? Collagen is still made, but it is not. It is fragile collagen. It is not tensile strength. It's a weak collagen. Weak collagen cannot hold the teeth properly. Even when you do brush, even gums cannot be held in position properly and they're slightly injured and infections go in and you develop gingivitis, inflammation of the gums and teeth may fall off. Even the collagen which make the support for all the microcirculation, microcirculation capillaries, very small blood vessels which are called capillaries. Capillaries are just tubes made of endothelial cells with some basement membrane or collagen around them. And when vitamin C deficiency is there, capillaries don't have a proper supporting collagen. Capillaries become very fragile and they rupture at many points. Is that right? Even during the wound, if you get some wound, injury, whenever you have some injury, blood clot is there. In the clot, fibroblasts come. Normally fibroblasts come in the wound area and they lay down collagen there. Collagen will be made good in vitamin C deficiency or bad? Bad. So what happens when vitamin C deficiency is there? Gums are injured, tooth may fall away, teeth may fall off, multiple micro hemorrhages occur from small vessels which are not supported. They are supported by, we call it capillaries become very fragile, right? Even wounds healing is very impaired. And if you don't replace this, and there's very severe deficiency of vitamin C for very long, even death of the person may occur. This whole disease is called scurvy. The whole disease is called scurvy. So you understand what is the relationship of scurvy with the molecular level? That scurvy is a clinical problem, set of clinical problems which occur because there's severe vitamin C deficiency, and due to that reason, one very important complication is that vitamin C acts as a cofactor for these two enzymes. Vitamin C dependent hydroxylases don't do hydroxylation of proline and lysine residues in alpha chains of collagen. And these alpha chains are made, even they make a triple helix, but because the side hooks are not there, so they don't get proper strength. Is that right? And collagen is weak. Am I clear? There's no problem up to this. Right. Now, collagen is still molecule is moving. Now it has become having multiple hydroxylations. Is that right? Now, after that, this molecule still moves further. At this step, it was hydroxylated. Next step, another area will come. And there's another enzyme there. And this enzyme, look at the face of the enzyme. Is it sweet face or not? It's a sweet face, isn't it?
That's another enzyme?
Yeah, this is another enzyme. Sweet face enzyme. Right, what it is going to do? It's going to add, it's a sweet face, it's going to add sugars on this molecule. Is that right? Especially glucose, right, or galactose. So we can say the name of this enzyme may be something like it is glucose transferase or galactose transferase. So what really it doing, that it will transfer to this peptide chain. What is this peptide chain called? Pro alpha peptide. It already had what things there? Hydroxyls, right? And out of these hydroxylations, some are proline, hydroxyprolines, some are hydroxylysines. It loves to add sugars on hydroxylysines. These sugars are especially monosaccharides. It may be glucose or it may be galactose.
Only on the lysine.
Hydroxylysine,
Not on the.
not on others. Right? There are special points where we should put the sugar. Sugar should be put in your mouth but not in your ear. It's the same way. So this enzyme knows where to put the sugar. It knows the appropriate places. The appropriate place to put the sugar on the peptide is hydroxylysine. Whole this process is called glycosylation. So what was happening? Initially this went over here. What was the process? Hydroxylation. Hydroxylation, I don't know, I love to teach. And what is this process called? Glycosylation. Is that right? No problem into this. And when once molecules are sugared, sweet, they love to come to each other. Isn't it? Now what happened? By this time, there's not only one peptide chain moving, maybe multiple peptide chains are moving through this process. Now they are hydroxylated and they are glycosylated. So they love to come near to each other. They are ready to make triple helix. You get it? Before making a good triple helix, you should have two things. You should be sweet and you should have the hooks. Is that right? Molecules know these things. Yes. Now they are ready for that action. Right? Now what happens? Before that, one thing needs to be done. Look, this is the peptide chain, right? Of course, like all peptide chains, it should have one carboxyl and another amino end. There's no fun in telling you people know these things. Is that right? Yeah. Now, this is one chain. Let's suppose here is one more chain. Of course, you cannot make the triple helix with one chain. There's another chain. They may be similar chains or they may be different. For example, all of them may be alpha 1, alpha 1, alpha 1, or it may be alpha 1, it may be alpha 2, it may be alpha 1, or any other combination. But I'm just showing here 3 chains. Should I show by different colors or same colors? No, but in the long run you will get trouble. Someone thinks there's something. But I don't know what is threesome. You don't want to know it. Who doesn't want to know it?
Let's keep this clean.
No, you think it's getting dirty? I don't know. I don't know what's going on in his mind. My God.
You shouldn't be teaching.
Yeah?
You shouldn't be teaching.
No, but actually look, he is not thinking about the collagen, he is thinking other things. This is the bad thing. Yeah. Now listen. What is happening? We are going to get what? Don't call it threesome, just call that the three peptide getting ready to intertwine. Now, they will start from which end? From carboxyl ends or from amino ends? Why you believe that? They should start, actually from one end, three of them will come together and they start twisting. And then that twisting should continue throughout. Actually, they start twisting from the carboxyl end. Actually, how it starts? Actually, the amino acids which are at the carboxyl end especially, and some amino end, they get sulfides, disulfide bonds. There are some cysteine amino acids which love to bind the sulfide. So they get disulfide bond here, also sulfide bonds form here. As they are more made on which end? Carboxyl end. So three carboxyl end come near to each other. Don't forget, all of them are having hydroxylation hooks and all of them having sweet points which are glycosylation points. And now some sulfide bonds form and then carboxyl end come near to each other. And then what you will find very soon. Right, so don't forget the cell was fibroblast. Right. Now what will happen in the next step? All this is travel going on in which? What thing? Endoplasmic reticulum. So what you will find? That what is this? Carboxyl end, right? And this carboxyl end of other chain, they intermingle with each other. And this is also, I don't know, they have started something. All of them have come together on this end, carboxyl end. And after that, the real action starts. They start twisting around each other, right? So what you will find that yes, this should be the blue. Now, here these are which ends? What are these ends? Amino ends, right? These are loose ends. What are these? Loose ends. So what you really see, there are also loose ends on this side, and these loose ends are it means this area is not making triple helix and this area is not making triple helix. Triple helix is made in the central portion. Is that right? It is just like that. You took the three ropes and you twist them in the central area and you leave the loose ends in the beginning and at the end. You're getting it? So it becomes something like this. Now this triple helix is formed. But in the end, we have loose ends. These ends are not making triple helix. Thank you. Is that right? This molecule, of course, the things which help in this triple helix, proper triple helix formation, number one, you needed glycine at every third position. Number two, you needed proper hydroxylation and glycosylation. Is that right? Now this triple helix is formed. Now this molecule is called, which molecule? This is called, 3 alpha peptide have made a triple helix. This is called collagen molecule. This is called collagen molecule, or it is simply called pro-collagen. Don't confuse pro-collagen with pro-alpha peptide. Come back. Pro-collagen is triple helix. It's the threesome. But when you come to pro-alpha peptide, it was just alpha peptide which has successfully entered into endoplasmic reticulum and cleaved away lost its signal sequence. And pro-alpha peptide undergoes through hydroxylation and then glycosylation, and then sulfides are formed in the end, and these help from carboxyl end, they come together, and zipper system start from this end towards that end. I mean, they start twisting from this end, and gradually this triple helix formation extends from carboxyl end to the amino end. But in the very beginning and very end, the loose ends are left. Yes.
The sulfide that you said was on the carboxyl end. That's what causes them
to twist.
To cause the triple helix. Okay.
Yeah. Is that right? Now, this is collagen molecule, or it is simply called what thing? Pro-collagen. So this is right. Now, this pro-collagen from endoplasmic reticulum, right, is now this pro-collagen is packed into some vesicles, and then this vesicle breaks away from the endoplasmic reticulum. It is sitting here, right? I will make it now with one color. This is pro-collagen molecule. Now it moves from endoplasmic reticulum, it makes a vesicle, it buds out, and this vesicle is called transfer vesicles. Transfer vesicles. Right. So where it is being transferred now? Not outside the cell. Before it has to go to that immigration department, get the visa. Yeah. There is an department from where it gets the visa and permission, because its name should not be in exit control list, right? So there's a very special arrangement here. This apparatus is called Golgi apparatus. So from the endoplasmic reticulum, transfer vesicles bud off and fuse with Golgi apparatus and pump these pro-collagen molecules into Golgi apparatus. In the Golgi apparatus, they put more sugar on it. You know, this collagen is going out to provide the strength. So they want to send it happily, right? So they add to it special sugar molecules to it. And these are not monosaccharides, they are multiple monosaccharides, so we call it oligosaccharides. So this oligosaccharide attachment here should not be confused with this glycosylation. Is that right? So again, further oligosaccharides are attached to it. Now it is ready to be expelled, right? Then what happens? Once sugars are attached with it, oligosaccharide, it has permission to go out. Then what happens? From here a small bud will come out. Is that right? This was transfer vesicle, and this is the secretory vesicle, because it will lead to secretion of it, right? So now it is packed into secretory vesicle. right? And secretory vesicles will, there are special arrangement for it, transport mechanisms. There are special microtubules and microfilaments, right? So that the secretory vesicle bind with them and then roll towards surface of the cell, because they don't want it get lost back somewhere. You are getting it? So what happens is secretory vesicles bind with these microtubules and microfilaments, and along them they start rolling towards the cell surface of the cell membrane, and eventually they come to the surface of cell membrane and open. The vesicular membrane and cell membrane fuse, and then this will jump out. What was this? Procollagen molecule. Is this loose ends, right? All this was intracellular steps in collagen synthesis. What we have brought out? Procollagen. Yet we have not made the collagen fibrils. There are further changes. There will be some enzymes dangling here to deal with it. We'll talk about that after the break.
I haven't